All right, so for our next section here, we're going to start off looking at the overall process of DNA replication. And so, to put this all in perspective, what we need to look at or think about again is why DNA needs to be replicated in the first place. And so, DNA replication is always going to happen before cell division, because remember, the ultimate goal of cell division is to produce identically or genetically identical cells. So to have that take place, DNA has to be copied, and then once that happens, the identical copies of DNA can be partitioned off into the two daughter cells, with them being completely genetically identical in the end. So, what we're doing next is getting into the details of DNA replication. Now, with DNA replication, and really the same thing is true of both transcription and translation, we're going to have three main step names. We're going to have initiation, which is where the process starts. We're going to have elongation, which is where something is going to be constructed. In the case of DNA replication, we're going to be building DNA. Transcription translation will be different as far as what we're building, but they also have their own respective elongation phases. And then we're also going to have a termination phase, and that's where the process is going to stop. So, to start off DNA replication with initiation, the first thing we're going to look at is the separation of the parent DNA strands. The parent DNA strands are the original strands that we're starting with and the ones that are going to be copied. So the two parent DNA strands are going to be unwound from each other, meaning that the hydrogen bonds between their complementary bases are broken using energy, and that will then separate the two DNA strands. Elongation, like I said, is where we're going to have the building of the new DNA daughter molecules from the parent strands. And this is primarily going to occur through the help of an enzyme called DNA polymerase, which is the main enzyme that does the bulk of the work when it comes to DNA replication. Eventually, termination takes place, and with DNA replication, termination is very quick and very straightforward. With the other processes, there's a bit more involvement there as far as what takes place with transcription or translation, but the same basic idea applies in that the processes stop with termination. All right, so this image here, we're actually looking at the beginnings of initiation. We have our parent DNA strands are blue in this picture, and they're being separated right here. So this GC used to be connected via three hydrogen bonds. Same thing with this GC. A and T would have been connected by two hydrogen bonds, and so forth. But all of those bonds have been broken, and now what we're doing is bringing in new nucleotides on these red strands and pairing them up with the old strand to create identical daughter strands. So we'll see how this all works and how this fits together. Now, DNA polymerase, which again is the main enzyme that does the bulk of the work of DNA replication, it turns out there really isn't just one DNA polymerase, there's actually three of them, but we're going to pretend that there's just one for our purposes. DNA polymerase is the enzyme that comes in and matches up the new DNA bases that are complementary to the old DNA bases. So, in other words, on the DNA strand, the old parent strand, if the old strand has an A on it, the new strand will have a T on it. If the old one has a G on it, the new one has a C. If the old one has a T, the new one has an A, and so on, right? You get the idea. So we're just bringing in complementary bases that match up with the bases on the old parental DNA strands, the original strands. Now, DNA polymerase, actually all of them, have a couple of unique features that we need to talk about here. The first thing to talk about is that DNA polymerases can only read in the 3' to 5' direction. And because they're reading in the 3' to 5' direction, that means they can only build in the 5' to 3' direction. So that's going to create a little bit of an obstacle that we have to overcome to understand how this actually works based on the fact these strands are running anti-parallel. So if you remember, anti-parallel means that if we're looking at the 3' end of one strand, the other complementary strand has to be a 5' end at that same location. So 3 goes with 5, 5 goes with 3, like we see up here on these pictures. This 5' end matches up with this 3' end, and that 5' matches up with that 3'. All right, so DNA polymerases can only read 3 to 5 and build 5' to 3'. The other thing about DNA polymerase to consider here is that DNA polymerase has to have an RNA primer in order to know where to start. In order for it to know where it needs to start synthesizing new DNA, it must begin with an RNA primer. And so our next enzyme that we'll talk about is DNA primase. DNA primase is an enzyme that comes in and creates an RNA primer. DNA primase is an RNA polymerase of a sort, which creates the RNA primer, and that RNA primer tells DNA polymerase where it's going to start constructing the new DNA. From that point on, it's very, very straightforward. All of the elongation involves is just bringing in bases that are complementary to the old bases on the original strands. From that point on, it's very, very straightforward. Another thing to consider here is that when we're unwinding the parental DNA, there's again a bit of a challenge here potentially, in that what we could eventually see is a degree of supercoiling that could take place. So in this picture up here, we see DNA strands have been separated from each other, but what's occurring is that downstream from that separation, the DNA strands are supercoiling. They're actually creating additional, like, second and third degree coils, which could eventually cause the DNA to become knotted and essentially unreadable at that point. So as the DNA strands are unwound by DNA polymerase, the strands are then stabilized by DNA helicase. DNA helicase, which has separated the strands, is then succeeded by DNA gyrase, which makes the strands stable. So whereas these supercoils are forming over here because of DNA helicase separating the parent DNA strands, down here the strands have been separated by helicase, but DNA gyrase has come in and is stabilizing the strands so that we do not see that supercoiling taking place. DNA gyrase stabilizes the parent strands so that we do not see that supercoiling taking place. We also are going to see what are called single-stranded binding proteins, or SSBs for short. Single-stranded binding proteins are there to help stabilize the nitrogenous bases on the nucleotides while the two complementary strands are separated from each other. So in other words, if we just jump back a couple of slides here, a little bit more actually right here. So wherever we have a G and C where the bond has been broken between them, this G and C could potentially be very reactive with other things in the environment. So the SSBs are going to come in and they're going to stabilize this guanine. They're going to stabilize this cytosine so that they do not react with other things in a way that we don't want them to. And they can only be paired up with their own new complementary bases. So G will be paired up with a C and nothing else because it's being stabilized by those single-stranded binding proteins. So there's a lot goes into this kind of early stages of replication between helicase separating the strands and gyrase stabilizing the strands and single-stranded binding proteins keeping the nucleotides from reacting with somebody that we don't want them to react to. It's a fairly complex little process, right, to make this early stage here work and take place properly. Now, the model of DNA replication that we're going to look at is called semi-discontinuous. So if we break down that word and think about what it means, right? If something is discontinuous, that means it occurs in separate pieces. If something is continuous, it means it occurs all in one piece. So something that's semi-discontinuous is somewhere kind of in the middle. And really what that means is that on the old parental original DNA strands, one of them will be synthesized continuously, the other one will be synthesized discontinuously. And so overall then, if we look at both strands taken together, what we end up with is a model of DNA replication that we describe as being semi-discontinuous. And the whole reason this takes place, the whole reason why it's semi-discontinuous relates back to the fact that the DNA polymerases can only read 3' to 5' and build 5' to 3'. That's what creates the need for this semi-discontinuous method of DNA replication. So we're going to have here two strands that are created. Both of these strands are the new strands, the daughter strands of the original parent strands. These two strands are going to be classified either as a leading strand, and the leading strand is the one that's made continuously, or a lagging strand, and the lagging strand is one that's created discontinuously. So again, if you take the continuous plus the discontinuous, you end up with semi-discontinuous. And so we're going to see why that occurs here in a second, but it all goes back to this whole idea of DNA polymerase only being able to read in one particular direction. So in this picture, this actually shows it as well as I think any image that I have. This is our old DNA double helix here. In this old DNA double helix, on the top, it's got 5'. At the other end, it's got 3' there. At the bottom of this, we have 3' at this end, matching up with a 5' on the first strand, and then going to the 5' end down here. So 5 with 3, 5 with 3, because of the anti-parallel nature of the strands. So if we take DNA helicase and separate those two parent DNA strands, we create what's called a replication fork. Now remember, the other thing about DNA polymerase, as far as is kind of unique aspects, in addition to only being able to read in one direction, DNA polymerase has to have an RNA primer to know where it has to start replicating things. So this yellow bit on the new strand over here is a primer of RNA that was created by DNA primase. And up here, same thing. This is a primer of RNA that was created by DNA primase. And so once these RNA primers are in place, once they've been created by DNA primase, then DNA polymerase can come in and start synthesizing new DNA. So on this new strand, the yellow is an RNA primer, and the blue is the new DNA. Same thing down here. The yellow is an RNA primer, and the blue is newly created DNA. All right, so this arrow, this new DNA is being synthesized by DNA polymerase. But the reason why it's semi-discontinuous is this, right? This bottom strand down here is running 3' to 5' at the other end. And as we said before, DNA polymerase has absolutely no problem reading 3 to 5. And therefore, if it's reading 3 to 5, we know it's also building 5' to 3' on the new strand. So that works if you're DNA polymerase. But look what's happening up here. When we create this replication fork, this top strand is reading 5 to 3. And DNA polymerase can't read 5 to 3, it can only read 3 to 5. So what happens is we put our RNA primer down here, and then we build backwards. Because if we build this section backwards, we are reading 3 to 5 in this direction, and building 5 to 3 in that direction, and that satisfies the rules of DNA polymerase. So this first fragment here, this first piece that we've built, is called an Okazaki fragment. Okazaki fragments are the pieces that are built of the lagging strand that are eventually tied together to create a fully functional DNA daughter strand. The leading strand is the one that's built in just one piece. This one, you've got one primer, and then you just keep synthesizing all the way to the end. You don't have to put any more primers in because this entire strand is reading 3 to 5 and building 5 to 3. Now, this Okazaki fragment up here is the same as the one up top. So we've now opened up the replication fork a bit more using our DNA helicase. We've stabilized it using DNA gyrase and single-stranded binding proteins. Now what we need to do is create another RNA primer here, and then build the next Okazaki fragment moving out away from the replication fork. Again, reading 3 to 5, building 5 to 3. And this just keeps going. This is going to occur. Remember, an average size piece of DNA is like 140 billion nucleotides. This just keeps happening until you've completely replicated every single piece of DNA in a cell. And it actually happens really, really fast. As long as it takes us to describe it, it takes us much less time to actually do this in real life. So that's kind of the basic idea here. We've got our kind of a hybrid molecule where we've got both DNA and RNA. We've got old DNA in green. We've got new DNA in blue. We've got yellow RNA primers. But really what we need to have is just the blue and green stuff. Right? We want the old DNA strand. We want the complementary new DNA daughter strand. But what we don't want in here is this RNA. So we're going to actually have to come back and remove that RNA primer and replace it with DNA. And that's actually one of the jobs of the sort of secondary DNA polymerases is removing the RNA primer and filling it in with DNA. So it's actually an entirely DNA molecule. So this is another kind of view of what's going on, which shows us our last enzyme we need to think about when we're talking about DNA replication, and that's going to be DNA ligase. So down here, we've got our leading strand. This bottom strand, we know would have been going 3' to 5'. We know that because this is a 3' up here, so this had to be 5'. If this is 5', then this had to be 3'. So this bottom strand is reading 3' to 5', which means we'd build it 5' to 3'. And then this top strand up here, this daughter strand is, this parent strand is going to be used to create a daughter strand that's made of Okazaki fragments. So originally, when this replication fork first formed, there was an RNA primer placed here on the original strand. And then from there, DNA polymerase synthesized a new piece of DNA in the 5' to 3' direction. And then we opened up the fork some more. New RNA primer was made, new DNA was made, and we then open up the replication fork a bit more. We have a new primer, we're going to create a new piece of DNA, and so forth. Now, the only other kind of wrinkle in all of this is the fact that once we take out these RNA primers and fill them in with DNA, we're going to get just a little bit of a nick right there in the backbone. There's a little break in the backbone here, and there's going to be one here, and there'll be one there, and there'll be one eventually here as well, because of how DNA polymerase removes the RNA primer and synthesizes in new DNA. There's always going to be a little bit of a break there in the backbone where you're missing phosphodiester bonds that normally link nucleotides together. So this enzyme up here, DNA ligase, its job is to come in and create a new phosphodiester bond here where the previous RNA primer had been removed and the new DNA had been synthesized. This break is then sealed by DNA ligase, and the same with all the others that are going to occur all the way on down the line with this piece of DNA. All right, so that's the basic idea with DNA replication. What we're going to do next in the last section of the video is I'm going to kind of wrap up by talking about mistakes that are made in DNA replication. We're going to see how frequent those are and kind of wrap up from there.